Hi folks, Maurice Gleeson from MySog here. The audio on this video improves after the first 30 seconds, so please bear with it.
What I want to talk about today is how we can go about trying to link the genealogical DNA tests that we can do to people from the past, and particularly identify ancestors that we might want to actually directly test. So I'll start with two of my ancestors. Maria Clark was born Maria Hurd, is my 3 times great grandmother. She's buried in the Anglican churchyard in Chickenham Holt in Devon. Her brother, William Hurd, is buried in the same village but in the graveyard of the chapel of the Brethren Chapel there. So this is not Church of England. And as I go on to explain, that creates some differences between what we want to do, need to do, if we want to do the best of their dialogue. So before I go into the details of that, I'm just going to go very quickly through the different types of DNA tests for genealogy and think about how far back we can reach with the different types of tests and connecting them to skeletons in the past. So basically I'm talking about 3 types of tests. The Y-DNA test in blue here tests the paternal line of a man going back in time, and you can only look at that male line going back one father after another. The mitochondrial DNA test will take you back through the maternal line to your mother's mother and her mother and so on. And then an autosomal DNA test reveals something about all of your ancestors going back a few generations. But after a few generations, not all of your ancestors. You don't get autosomal DNA from all of your ancestors, only from a fraction of them. And that limits what we can do with autosomal DNA comparing to people from the past. So that's the basic framework. I'll think about how we might use each of these types of genetic evidence to connect people from the past. If we do autosomal DNA matching, then with the current sort of methods that you can buy kits for in various stores around the hall here, you're guaranteed that if you compare your DNA with somebody who's a second cousin, you'll get a match. If you go beyond the second cousin, then some of your relatives won't match and some will. So by the time you get out to fourth cousins, you'll only match about half of your fourth cousins, and half of them you won't share DNA with. Now, in terms of looking back in the past, we need to think about the degree of genetic relatedness. So a second cousin is a 5th degree genetic relative, and a fourth cousin is a 9th degree relative. So if you project that back in the past, in a direct ancestral line, a 5th degree relative is your 3 times great grandparents. And a 9th degree, going back in the direct line, somebody who is 9 degrees separated is your 7 times great grandparents. So with an average generation of about 30 years, 3 times great grandparents could take you about 150 years from the birth of the person that you're testing, and a 7 times great grandparent might take you about 270 to 280 years from the person that you're testing. But obviously, so if you could locate your 3 times great grandparents, like that gravestone I just showed you with my 3 times great grandmother, you could guarantee that you share DNA with them, and if you can extract DNA, you'll get a match. You go back another 4 generations, then only half of those 7 times great grandparents will share DNA with you. And so you'll, even if you could do the perfect retrieval of the skeleton and extract DNA, there's only a 50% chance you get a match with autosomal DNA. So it runs out of steam as you go back in time. So here with Maria Clark, I'd have a 100% chance of an autosomal match. With her brother, I'd have greater than 99% chance, but there would still be a small chance that it wouldn't match at all. And if I went back further generations, those numbers would go down. With Y chromosome DNA going back in the male line, we can look either at the STRs, so if you go and get a test for your Y DNA from family tree DNA, the STRs or microsatellites will be what they would test, and you get a 37 marker test or a 67 marker test, whatever it is. They're useful for genealogy because they have a relatively high mutation rate in the panel of markers that are looked at, so you can detect close relatives and more distant relatives over genealogical timescales. The sort of tests that tend to be done with the SNPs, the individual mutations in the Y chromosome, have a lower rate because we don't test very many of them really in the sort of testing that's normally done. But with the STRs, the mutations can go back and forth in different directions, and so the connection isn't always clear who is actually closer than somebody else. Whereas with the SNPs, those mutations tend to happen just once, and everybody who has that mutation has a common ancestor. And then if somebody else shares another mutation but doesn't share the first one, there must be more distantly related. So you can always create a clearer phylogeny. Mark showed some of those phylogenies in his talk just now. So in terms of looking at DNA from ancestors, the STRs might be good for recent ancestors, but the SNPs will be better for more distant ancestors as you go back in time. For mitochondrial DNA, you're just looking at this internal line, which I'll come back to a bit later about what that implies in terms of genealogy. The mutation rate on mitochondrial DNA is relatively low, and some of the haplogroups that you would test are extremely common, particularly amongst Europeans. Some of them are very common, so you might, if you belong to one of those common haplogroups, then if you pick somebody else at random in Europe, you'll get a good chance of matching them, because you have some ancestor thousands and thousands of years ago. And that's not going to say anything about your genealogy over the last few hundred years. So there's a risk with mitochondrial DNA that you just get something uninformative. Okay, so that's what, in theory, we could do if we could get the perfect DNA sample out of one of our ancestors. How would we go about it? Well, first of all, you have to meet the legal requirements. I'm going to talk about England and Wales. There are similar but different rules in Scotland and in Northern Ireland and in Southern Ireland. And when you start going to other countries, they get even more different. So if you're going to excavate human remains in England or Wales, then you have to have an excavation licence from the Ministry of Justice. And it doesn't matter how you come across the human remains. If you find them accidentally and you want to take them out of the ground, you need a licence. And that licence will specify that you must remove the remains from the ground decently and respectfully, and you must specify what you're going to do with them after you've excavated them, whether you rebury them or whether they're deposited in a museum, and things like that. So wherever your burial is, you have to have an excavation licence. If you are excavating where the majority of people in the past are buried, and that's in the Church of England graveyard, then you also have to go through the church's legal process. So the church has its own separate legal process, and you have to have what's called a faculty from the local diocese in order to excavate and do any disturbance on consecrated ground. So if you have found the grave of one of your ancestors and want to excavate it, you have to persuade the Ministry of Justice that it's a good thing to do. That's probably not too difficult as long as you've got good reasons. You've also got to persuade the diocese of the Church of England that it's a good thing to do. But the Church of England will have a presumption that graves should lie undisturbed unless there's good reason to disturb them. So you've got to persuade those two groups of people that this is a good thing to do and get the permission. So going back to my two examples, if I wanted to excavate Maria Clark, I would have to get a Ministry of Justice licence and get permission from the Church of England. If I wanted to excavate William Hurd's grave, I had to get the Ministry of Justice licence, and I need the permission of the landowner. And if the landowner and the tenant of the land are different, you may need permission for both of them. But that's generally easier to do than getting permission from the Church of England. Though, depending on if it's a particular religious group, then they may have extreme objections to you doing anything with the grave at all. The Church of England and the Ministry of Justice would also expect you to abide by national guidelines for excavating graves. So there's a group called the Advisory Panel on the Archaeology of Burials in England. They provide guidelines on best practice in treating human remains excavated from Christian burial grounds. That's where most of our ancestors are going to be in this country. And also guidelines on what you should do if you're doing destructive analysis. So if you're going to do a genetic analysis, you're going to have to take a piece of bone, you're going to have to dissolve it and destroy it. So you will need to follow the guidelines of science in the game as well, about making sure that what you do is going to be scientifically productive, that it's minimally damaging, and so on. So these guidelines are on the web if you really need to see. And you see here that this is actually a reburial of medieval remains where the modern friars from the same order were present at the reburial. And that reburial will be the default for anything excavated from the Church of England graveyard. From other graveyards, it will depend on the Ministry of Justice license. So if you excavate from Church of England graveyard, the chances are that there will be quite a strict requirement to replace the human remains within the graveyard within a few years. Okay, so you jump through the legal hoops, you get permission to go and dig a hole in the ground. What else do you need to think about? The next thing you need to think about is what is actually there above ground before you start digging. So here's the tombstone again. Who's buried here? Well, is it not obvious it's Maria Clark? Well, maybe. What does the gravestone say? Sacred to the memory of Maria Clark. It doesn't say, here lies Maria Clark. So there are quite a lot of gravestones which commemorate more people than are actually in the grave. So you can't assume that when you see the name on a gravestone that actually that person is buried there. And usually it's not explicit that they're not buried there. I have seen one example on the cemetery I was working on in Houston in North Yorkshire, where the churchyard had been closed. There was a grave which was due, where a man was buried, his wife was due also to be buried in the same grave. Somebody messed up the legal process and the churchyard was closed. She couldn't be buried in the same grave as him. She's commemorated on the stone with a complaint about the legal process that prevented her being buried there. But she's buried several miles away. But that may not be apparent. So you have to be quite careful. That's not direct evidence of who's in the grave. The other thing is that stones do move. Stones get knocked over in graveyards, and then somebody puts them up again, and they may not go back in exactly the same place. And a lot of churchyards, you can go in, certainly the ones that I walk past on the way to work, you go into the churchyard, all the tombstones are neatly around the edge on the wall, so it's easy to mow the grass. Now, none of them tell you anything about where somebody is buried. So it's important to consider whether the stone might have moved. Was it in the right place initially? You can't put a stone up immediately after the grave is dug and filled. You have to wait for the soil to settle. So it may be months or even more than a year before the stone is erected. And in that time, in a graveyard where there aren't many other stones, it would be easy to get to the wrong place. And you also have to worry about whether the stone commemorates everybody in the grave. I mentioned that lots of stones commemorate people who aren't there, but some people get put in graves and don't get commemorated on the stone. And we know this from archaeological excavations in this churchyard, where you have a stone with two names on it, maybe. You excavate the grave, there are 3 or 4 people in it. And you don't know which of those 3 or 4 are the ones who commemorate and which ones aren't. So you have to worry about this, not actually direct evidence. And in many cases, there might not be a memorial stone anywhere. If you're lucky, then there's a plan of the graveyard that tells you where the plots are, but in most churchyards there won't be. You just have an entry in the burial register which tells you that there, somewhere, here's God's acre, and somewhere in the acre of ground. very difficult. Okay, so this this actually came up after I had devised the lecture. So you've probably heard, may have seen this in the news in the last month. This project where Leicester University were approached to see if they could identify Jack the Ripper's last victim. And the first thing you have to do is identify her grave. So there's been quite a lot of publicity for the idea that they're going to identify the victim. If you read right down to the bottom of this press release, then Jerry King says that the successful search for Ellen Kelly's remains will require an enormous effort. We don't know exactly where the grave is in the graveyard. We know a general area. We've had to excavate lots of graves. We don't know what state of preservation the remains are going to be in. So it actually becomes prohibitive simply to find the right grave before even doing anything with DNA. And of course, that sort of information, what you already know, has to go into your exhumation licence. The Ministry of Justice is not going to give you permission to dig up 30 people in order to find the one that you want. Okay, so let's assume that we can actually find the right grave and we can start digging. What will we find? Well, survival of the bones isn't guaranteed. What we hope is that we get some nice complete skeleton like this, neatly laid out in the grave, and one individual. That's really nice. No problems with sorting out multiple individuals in the grave, working out which bone belongs to which person. But in a lot of parts of Britain, the soils are acidic, the bones will dissolve. You excavate a grave like this and you'll just have an empty grave because the remains are completely decayed. And that can happen over a matter of 100 or 200 years. So again, at Houston, some of the graves we looked at, they're fragmentary. We might have parts of long bones, no ribs, and the other small bones will be disappeared, just fragments of the larger bones left. And those are 19th century burials, so they can go quite quickly. The other thing is disturbance in the graveyard. New graves are always being dug in graveyards, and there's always a risk that they'll disturb the grave that's already there. Especially if it's a graveyard which doesn't have markers in it, then a new grave may well cut through an old one, and in fact, that was routine practice until the use of headstones became very common. So you think about the scene from Hamlet, where the gravedigger is digging and he finds the skull of poor Yorick, and he actually knows who it is, and nobody's bothered that he's disturbing this grave and pushing it to one side to make a new space for another one. So that would be routine practice up until the point where you get permanent grave markers and there's a longer term memory of who's there and an idea that this is a permanent resting place that should never be disturbed. If you're unlucky, you get other sorts of disturbance as well. So one of my local cemeteries has a nice badger set in it. Every so often the badgers bring out bones, they're in the way. So you may not find a nice neat grave like this. You never know until you actually excavate it. When you get to excavate the grave, the best thing you can hope to find is a coffin plate. So from the 18th century onwards, it's quite common to have a metal plate on the top of the coffin which named the individual. If you've got that and it's neatly placed above the body, a few centimetres above the body, then you've got really good evidence of who that skeleton is. That's probably the best evidence you're going to get for the identity of an archaeological skeleton from the archaeological evidence. We have quite a lot of excavated skeletons where we know who they are. So a lot of the big excavations in London that have disturbed 18th and 19th century graveyards have produced coffin plates and named individuals. Quite a lot of them have been reburied, but some are still in the museum. We don't even have to do excavation for their ancestors already in the museum. After you've excavated, you need to make sure that the individual you've excavated actually matches all the other things you expect. Before you start spending lots of money on ancient DNA analysis, do some basic homework. Get an osteologist to look at the skeleton to tell you what age it is, what sex. If that doesn't match what you're expecting, then you might want to think again before you spend more money. And the other thing is, if you're going back beyond about 1700, then you may want to confirm the date of the skeleton as well by getting a radiocarbon date. And that's what I would do before I did anything else on skeletal collection, is make sure that I understand the skeleton that I've got and when it's from and what its biological characteristics are before actually analysing it. Okay, so you get a skeleton and you're going to do ancient DNA analysis. Now, all ancient DNA scientists look the same. Here they are. I don't think there's any of the ones who are sitting in this corner of the room. But these are all different people, working in different laboratories around the world. And I have no idea, without going back to Google and looking at the image, which each of these is. So, and why is this? Because our major enemy of ancient DNA is contamination from our own DNA. So the DNA that is going to be in the bones is going to be degraded, it's going to be easier, more easily destroyed than our own DNA, and one piece of your dandruff contains your complete genome, recently produced, not subject to decay in the archaeological record, and can make a big difference to your analysis of your ancient DNA. Ideally, we would actually take this back to the excavation stage as well, and we would excavate with controls to try and prevent contamination. And one of the things that really, when you think about if you're digging up your ancestors, is don't do it yourself. Because what you're expecting is that you share some DNA with these ancestors. But if you contaminate them, you're going to match the sample that you extract. So you have to employ somebody else to do it. But if you can avoid contamination, and it's not absolutely necessary now because we can actually distinguish contamination from the ancient stuff after we've done the analysis, but it's best not to have it there if you can help. You have to worry about the DNA that you get out. Okay, so here is, so your DNA in the chromosomes that are analysed in the genealogical test, there's millions of base pairs wrong in the measurement of how long DNA is. Here's a chart of the length of pieces of ancient DNA that come out of a typical ancient DNA analysis. This is perhaps a little on the short side. You may expect that the chart this way a bit more, especially if we're dealing with recent remains. But you can see this goes up to 165. Not million, just 165. And so these pieces are very, very small. And from these pieces, you have to reconstruct the jigsaw to reconstruct the genetic signal of the ancient person. That's a big computational job. You analyse all these pieces of DNA, you get the computer to reassemble them into a genome. But it's difficult if you want to use STRs. So if you're using STRs, so here's family tree DNA's 12 marker panel, the first 12, which will be in any other test that you do. They're basically short pieces of DNA that repeat, and the number of repeats you have is the marker that is used to estimate relatedness. So these are the motifs. You see that they tend to be 3 or 4 letters of the genetic code in length, and they repeat, well, between 6 and 30 times roughly. That means that if you want to know how many repeats you've got to have a piece of DNA that is complete with all those repeats in it. Because if you just have a fragment of it and it's got a series of repeats, you can't put the jigsaw together because you don't know where they're overlapping. So you need a piece of DNA this long, which contains the repeats, plus some DNA either side of it, so you can tell where you are in the genome. And that means that the pieces of DNA that you need to be 100, 200, 250 base pairs long. That means you're, on this chart, you're out here somewhere. Those size brackets of DNA are fairly rare in ancient DNA, and therefore it's not impossible, but it's going to be difficult to do STRs from ancient DNA. It's not impossible. If you throw enough effort and technical expertise, I think you can do it. It was done for Richard III, but that was a thing where they were determined to get a result, and they put an awful lot of effort into getting the results out of the ancient DNA. It's not something that you're going to do routinely. Okay, if we turn to the other form of y-DNA, we could look at the SNPs and the individual mutations. There's no commercial kit available to do degraded y-DNA for forensic or archaeological purposes, so your lab needs to make up its own method for doing this. So there's no routine way to do it. That's going to be a bit of a problem, but with a bit of method development we might come over it. So we would need new methods for analysing y-DNA to make connections into survey projects and things like that. It might be possible to use selected STRs, pick the short ones, be easier to analyse them. You get limited information and probably rule out some connections but not be sure about others. Or do some sort of bespoke testing where you test the SNPs in the archaeological specimens and you test them in the living individual, but not in the same way that any of the testing companies are doing them today. So you have to do your own genealogical testing and not the commercially available one. Ancient mitochondrial DNA, this is much easier. Mitochondrial DNA is present in many copies in the cell. The autosomal DNA, y-DNA, there's one copy in each cell, 2 copies in each autosome. Mitochondrial DNA, there are multiple copies. That means we're in a good starting point for our archaeological record. The DNA degrades, it decays, it gets lost, but we start out with many more copies of mitochondrial DNA, and therefore its chances of survival are much greater. That makes it easier to recover, and in a lot of ancient DNA studies, it's what you would analyse first just to test for survival of DNA. So it's what we've done on a couple of our Scottish soldiers. We don't talk about the Scottish soldiers very much, but we have done just some basic tests of mitochondrial DNA to test for survival of DNA. If you can't extract mitochondrial DNA from a sample, then there's not much point going for autosomal DNA because chances are it isn't there. And of course, mitochondrial DNA had a key role in identifying Richard III, and that was the direct genetic connection that could be made by a group and match the genealogy. But with Richard III, there's a slight element of luck that he has a fairly rare haplogroup. So the chances of it just being a random match, if you've got the genealogy wrong, and if you ended up with some random person because you made a mistake in your genealogy, then the chances are he wouldn't have matched some random person. If you had a common haplogroup, then it might be harder. Ancient autosomal DNA, okay, so as quickly as possible. There are several lectures today which have talked about analysing ancient autosomal DNA. The analysis is done for genealogical purposes. We need to be able to look at all the individual SNPs that are tested. And when we're covering ancient DNA, the coverage is variable. So sometimes we'll get good coverage and we'll get multiple analyses of the same point on the genome. We can be confident what it is. Other times, the average coverage will be less than one on each of the SNPs. So something we don't have at all, something we have multiple analyses of, for sure it's there. And that means that matching is difficult and you may not get distant relatives. So, okay, and you get the same sort of problem as you get comparing autosomal DNA tests from different companies when they start using divergent testing chips. People I know are complaining that if you transfer an ancestry version 2 kit family tree DNA, you don't get the list of very distant relatives because there's not enough overlap between the two sets of tests to actually identify distant relatives. So if I compare myself with an ancestry test to Hingston 2, who's an Anglo-Saxon individual from near Cambridge, so a typical ancient DNA sample. The ancestry test has got 700,000 snips in it, but the overlap with what we actually managed to recover from Hingston is only 300,000, less than 300,000, which is not terribly good. Whereas if I compare to some random person who's on GevMatch, who's standing there back there, comparing ancestry to family tree DNA, I get 600,000 snips, so I've got more than twice as many to compare as I had with the ancient sample. This is not a match. I don't match Morris at any sensible distance. I don't really match the Hingston one very well either. But then, that is 1,300 years ago. The degree of genetic relatedness ought to be enormous. There should be 40 generations in the past if I was a direct descendant. So that limits what we could do with matching then, is actually the recovery of the ancient DNA. So although I might go to my 3 times great grandmother and say, I definitely share DNA with her so I can identify the skeleton. If I can only recover half the snips that I need for the analysis, I may not be able to get out to that degree of relatedness. And then, of course, once you've got your genetic data, you also need to have your paper genealogy, and you need to check the records. And that introduces 2 things that we have to think about with trying to match ancient DNA to modern DNA. First of all, tracing that mitochondrial DNA line is going to be the easiest thing to do with the ancient DNA, but the female line changes surname every generation in the British genealogy, so the chance of making a mistake or coming up against the barrier where you can't determine what the maiden surname was is much greater. So establishing the genealogy that is going to match or not match your mitochondrial DNA is actually a lot more difficult than any other lineage which includes multiple males. And then when it comes to the Y DNA, we have to worry about non-paternity. So this is the issue that had Richard III. Okay, it's a high-profile lineage. You can trace the genealogical records really easy. The paper trail is very good, but it doesn't match. And in that lineage, there are at least 2 non-paternity events from the genetic analysis that's done for Richard III. So as you go back in time, we have to worry about, on average, perhaps a 2% chance in every generation. 2% doesn't sound very much. Go back 10 generations, then 2% is becoming a lot more significant probability. So the cumulative effect can be quite large. If you want to go back to medieval ancestors like Richard III, some of the famous medieval person, then you're talking about dozens of generations, and therefore a much more higher chance of an effect. Okay, so it's all possible, but there are lots of differences. The other thing you have to think about is how much is this going to cost? How deep are your pockets? So you're going to do an archaeological investigation of a grave. You're not going to get away with more than a few £1,000s for doing that excavation. It might cost you tens of thousands of pounds if it's in the wrong place. So that will actually probably be a substantial part of the cost of doing this. It's not the only cost. The next stage, I said, always do an archaeological report, do radiocarbon dating, maybe do screening for DNA preservation. Each of those is going to cost you several £100s. So we're talking about that's £1,000, £1,500, and you've still got the chance of failure. It may turn out not to be the person you think it is, and there's no point carrying on if you've got to the point where there's no DNA or it's not the person you think it is. And then you've got to actually do autosomal DNA and mitochondrial DNA or develop a method for Y-DNA. Developing a method for Y-DNA is not going to be cheap, because you can't just take a routine method and just have the costs of applying it. You've got to employ a researcher to work for several months to develop a method that is going to work. So I'm leaving Y-DNA out for the moment. For autosomal DNA or mitochondrial DNA, then it's probably going to cost you a few £1,000s per sample to actually do the analysis. So, the overall price, well, if it comes in at less than 5 figures, you're probably doing quite well. So that's, some of these costs may come down, and some of these costs may disappear in certain circumstances. So the cost of the sequencing is coming down. The cost of doing the work, avoiding contamination in an extra DNA lab is not going to change. You've still got to have a clean lab, which is very carefully kept clean. You've still got to have all the gear to do the extra DNA extraction. The sequencing itself is becoming cheaper and cheaper. And it may be you're lucky with the archaeological investigation. Maybe somebody's dug up your ancestry already and they're lying in a museum. You've got to pay for access to the museum, which is going to cost you millions, not just doing excavation. So, not necessarily that expensive, but that's the chances of it all. So, in conclusion, digging up your ancestors for DNA, it's possible, but it's not easy. Thank you very much.
Okay. Thanks very much, Andrew. Fantastic presentation. Okay, well, I think that certainly there's a lot of issues raised in that presentation about digging up ancestors, and I think everybody in the audience is probably thinking now, who in my family tree would I really like to dig up? So, who would like to start off the questions with their own particular family example of who they want to dig up and why? We have Gerard here in the front.
I wasn't anticipating your question, but I will answer. That would be the 8th century, the Eli O'Connor. We know exactly where he is buried. In fact, there's also a modern gravestone over the ancient burial. And he was digging up the Eli O'Connor. Now, one question. Europe's churches, monasteries, abbeys are stacked with ancient relics of old people. What's the potential of getting access to those?
To relics.
To relics.
Okay. There is a...
Like, for example, if you go to Auckland and look in that great big silver casket, is the skeleton of a giant called Charlemagne.
Yeah. There is actually a project which is trying to look at relics. And we've done, there was supposedly the bones of John the Baptist in a church in Bulgaria, and they've done dating, and it's the right date, and they've done some genetic analysis, and it's Middle Eastern. Whether that matches to John the Baptist, who knows? There's also been not genetic analysis, but a series of studies on saints' relics in Belgium. And when they radiocarbon date them, half of them are the right date for the saint and half of them aren't. And some of them are much older than the saint. So it appears that in some cases a Roman burial has been dug up and somebody has said, well, this is the saint, it's gone into a casket. But actually it's not your 9th century saint. So, and part of the difficulty, even going back to Eli O'Carroll example, will be the question of in whether it is 1200 years.
1888
Yeah,
was
so,
the number on the grave.
so 12 or 1300 years, what's the chance that it has not been disturbed? We know from the Barrymore example that has been mentioned already that that's an 18th century tomb which has been disturbed at least twice. And the disturbances weren't known about. So they went in expecting it to be intact.
Here's a suggestion. If you go to Iona Abbey,
Yes.
in the Isle of Iona, there are at least 50 remains of kings of Scotland, Ireland and Norway
Yes.
who are buried there, not necessarily in their right? Because there wasn't very good right place, or all of the inscriptions had eroded. So you could convince someone that it would be a good idea to dig them up and put them in the right place.
We have a question back here.
So obviously in an ideal situation, you would dig someone up and then they would go into a database like family tree DNA, so you can compare them to everyone. Is there any possibility of that ever being a thing?
With some of the ancient ones are on GEDmatch. So the reason I can compare easily to Hingston is because somebody has taken the ancient DNA data and processed it to find out which slips are there and create a file on GEDmatch. So that's just a computational issue of processing data, and you know how to do it, so it would be easy to set up something to do that, to get the ancient remains into the databases.
Who would you contact for that?
Who was the chap that actually did it on GEDmatch?
Felix Emmanuel?
Felix Emmanuel? Anybody know?
I think he stopped doing it. No, he's not doing it.
He's not doing it anymore.
But I think he's written up his methodology. So if you've got the skills to do the bioinformatic processing, then it wouldn't be difficult.
Probably the thing to do would be to go onto GEDmatch, search for Hingston, and see the email address associated with it. And you can get in touch with them and say, could you do me a favour? Question here at the front.
Moving on from people who might like to dig up to people that have actually been dug up, the Talensa Valley battle site seems to have produced a large number of bodies where we're told that the DNA is in good condition and probably recoverable. Do we have any information as to when that information is available?
I don't know who's working on that. Mark might know. Do you know who's working on the battle site in Poland?
No, I'm not sure. But how long ago was it? So how many generations ago?
It's a Bronze Age battle site.
Okay. Yeah. No, I'm not sure about that.
They've got a mass grave.
But as you point out, it's been worked out anyway. If you go back 10 generations, when your ancestors 10 generations ago, you've only got a 0.1% chance of carrying any of their DNA at all.
But if you could do it live,
Yeah.
it's past it.
We're told that we've covered over 100 bodies and the DNA is in good condition. I don't know that, but that's what's been reported. Now, if that's the case, that's going to considerably change the pattern of ancient remains, is it not?
Yeah, I mean, there are about 350 ancient DNA remains from Europe at the moment floating around, or thereabouts. So, yeah, it will change it a lot. Yeah, but it'll change it a lot in one particular place, right?
And it's not going to help you with your genealogy.
Yeah.
Because it's so far in the past.
Question here from Debbie.
Recently, I read in the papers that there was that 60,000 bodies have been uncovered as part of the HS2 excavations.
Well, they will be, yes.
So, I mean, of course, there must be, everyone probably will have an ancestor there, but the cost of actually doing DNA testing on 60,000 samples would be astronomical. Is that something that ever might happen?
Well, that was Dan Bradley's vision in his talk, was that he would be able to take the data and shake it up, attach it to a USB device on your computer and get it. So in principle, well, I guess costs are coming down and techniques are being refined, so it may be possible. But I don't see that. I think the cost will come down to similar to a radiocarbon date. It will be a few £100s to do at best. The cost of radiocarbon dates today is still the same as when I started using them 25 years ago. They're still about £400. £400. The technology's improved, the better quality dates, but the cost is about the same as it was. So I think the genetic costs are actually still falling, but they're reached a point where
we have a question here as well. So.
First of all, can I say how wonderful it is to have an academic here?
That's quite a few.
The second is a really flippant question. I am getting near the point where my DNA is ancient. Is it worth preserving me so that when technology permits, they're able to recreate me? And my second question is, will I have the experiences that I've had in this life?
I don't think, as they tried to recreate you, so much of what has shaped you has been your environment. So the DNA gives a framework, but you've been shaped very much by your environment. So you might not look, you will look similar, but you might not be the same.
Yeah, the question is, the question is, would you be creating, given it was possible to do it, would you be creating an identical twin, or something just a bit more than an identical twin? There is some suggestion that you'd be creating something just a bit more than an identical twin, because there's a process called the epigenetic imprinting, which can affect the way your genes are expressed based upon your life experiences. So, for example, if you went through a lot of starvation in your life, then they made a clone of you, which would be equivalent to your twin, but it would be your twin that would be a bit smaller.
It's funny this conversation has come up because this morning we were talking about how there is actually a GoFundMe campaign to reconstruct the genome of Joseph Smith Jr., who was the founder of the Mormon Church. It's not so that we can clone him, it's so that we can prove that he didn't actually have children with his, what are called his, they were his legal wife and his sealed wife. His legal wife, he had lots of descendants with. The sealed wives were just polygamous wives that he kind of had a spiritual connection with. But if we did reconstruct this genome of Joseph Smith, would we be able to then take a newly fertilized egg, use CRISPR to snip out the snips that we don't want and put in the Joseph Smith snips that we do want, and produce Joseph Smith?
If you wanted a Joseph Smith, I mean, CRISPR technology is not that advanced. I mean, you can put a few things in there anyway. But this is Thomas Jefferson all over again, isn't it, really?
And we're doing Thomas Jefferson's descendants as well. So there's a big project to look at all the descendants of Thomas Jefferson. I've recreated my mother's genome, 75% of it, using Lazarus functional get match, and a whole load of my maternal cousins. But theoretically, you could keep on doing Lazarus generation by generation until you get back to Joseph Smith.
But the problem is you 75% from one generation to the next, you 75% in the next generation, and 75% in the next generation, and you're soon into the 10%.
But you know, I mean, you can follow that reasoning. So, in fact, each person here has got about 1.5% Neanderthal ancestry. But the point is that we've each got different Neanderthal ancestry. So, if we did some back-of-an-envelope calculations recently. It would take about 30 generations if we bred people in a highly directed way, an extremely unethical way, to that we might be able to get somebody up to about 50% Neanderthal after about 30 generations of highly directed and unethical breeding.
That might not be a bad thing, because apparently they were more intelligent than humans, but unfortunately they looked like Scottish people. So maybe that's a good place to stop the conversation. And can I just say a big thank you to Andrew Millard for a fantastic presentation. Thank you very, very much.
So sorry to trouble you.
No problem.
Your colleague suggested I have a very quick chat with you.
Sure.